Hello, everyone, and welcome to today's AAPS eChalk Talk, partnered by dsm-firmenich. You may submit your questions at any time by entering them into the Questions tab of the sidebar. It is my pleasure to introduce today's moderator, Dr. Fernanda Onofre.
Thank you, Stacy. And many thanks to AAPS for the opportunity to present here today and for the chance to discuss with all of you and the audience about this important topic of nitrosamines. And before I pass the microphone to our presenters, I want to briefly introduce dsm-firmenich for those of you in the audience that are not so familiar with our company. dsm-firmenich was formed in May of this year as a result of the merger of DSM and Firmenich, two powerhouses that combine their portfolio solutions for customers that include innovative nutrients, flavors, and fragrances to various markets. In the pharma solution segment, we offer vitamins, carotenoids, CBD APIs, lipids, as well as specialty excipients to the pharmaceutical industry. And we're going to do a deep dive today on specialty excipients and how the vitamins can play a vital role as a critical excipient for nitrosamine mitigation in drug products. And to help you navigate this very complex world of nitrosamines, I have Anne-Cécile Bain and Zdravka Micic with me today. Zdravka has a PhD in pharmaceutical sciences and leads various projects on the role of vitamins and other nutritional actives in oral solid dosage forms. And Cecile has a PhD in molecular pharmacology and toxicology and leads global innovation projects to evaluate vitamins and nutritional lipids in the pharma and medical nutrition segments. So without any further delay, Zdravka and Anne-Cécile, the microphone is yours.
Thank you, Fernanda. Hello, everyone, and thank you for attending this e-learning session. The objectives of today's session are, one, to share some information about nitrite scavengers of use for nitrosamine mitigation, and two, to explore key aspects when formulating with ascorbic acid and alpha-tocopherol. If you go to the next slide, so you'll see that nitrosamines are formed by a chemical reaction between a nitrosating agent and a secondary or tertiary amine. A number of these compounds have carcinogenic potential, and that's been known and studied for decades. In fact, the first link of nitrosamine with cancer dates back to the 50s, when a couple of UK scientists reported NDMA as a carcinogen in an animal model. But after N-nitrosamine contaminants were detected in pharmaceutical products in 2018, regulatory authorities set a framework for the risk assessment, the testing, and the mitigation of N-nitrosamine in drug products. Now, this development really sparked a new level of interest in ways to mitigate the formation of these unwanted compounds. And the condition for nitrosamine formation can be met at many steps during the production of drug product, from the drug substance themselves to the excipients, the solvents, the water that's used, the manufacturing itself, the packaging, and the storage. All can create conditions for the generation of nitrosamines. Now, many strategies can be used to deal with nitrosamine formation. For example, and lots of you will be familiar with this, there are strong qualification programs for all compounds used in the manufacturing, trying to keep the level of nitrosating precursors as low as possible. And today, we're going to focus on one of the strategies drug developers can use to reformulate existing drugs and develop their new drug, and that is the use of nitrite scavengers to reduce and stop nitrosamines from being formed. And what you see on this slide is a list of nitrite scavengers. There's a lot of them able to do the job, and so you see some of them listed on this slide, but they're not all equal, however. Some of them are more active than others, or are more appropriate for one form over another. Some of them don't appear in the inactive ingredient database of the FDA, and as such are not necessarily as attractive. Others are active in solution, but don't appear to work in solid. So two of them which have consistently given good results for nitrite scavenging abilities are ascorbic acid in particular and alpha-tocopherol. Now, since results that have been obtained over the year, if you move on to the next slide. Yes, you can see on this slide, which were results obtained by Dr. Nenda and his team in 2021. And it was a very important result, because they looked at the inhibition efficiency of the number of nitrite scavengers, among them ascorbic acid, sodium ascorbate, alpha-tocopherol, but as well as ferulic acid and caffeic acid in a solid drug model. And they saw that these molecules had efficacy in reducing nitrosamines being formed, even in that solid model, and that was very exciting to see. Now you can see here that caffeic acid was as active as ascorbic. Caffeic acid was as active as ascorbic acid, but caffeic acid, of course, is not on the inactive ingredient database. So again, ascorbic acid, alpha-tocopherol, those kind of molecules seem to be working even in solid forms. So when you go to the next slide, you will see also some further evidence that built since Nenda published his study in 2021. There's a paper by Omchak and his team looking at a variety of nitrite scavengers. It was 19 in solution, and then some of them in tablets, really showing again that it's not just a one experiment, one observation. It has the ability to work also in tablet forms. Recently, in June, the FDA hosted a workshop on the topic of nitrosamines, and Dr. Shaklila showed that ascorbic acid, again, decreased the formation of nitrosamine in drug products effectively. Just recently, 2 weeks ago, we had Dr. Wong at the pharma meeting in the UK, showing again that ascorbic acid was very effective in a tablet form, and in her case, she was comparing to amino acids and to an amino acid, and that was again more effective. Beyond this, so it looks like there is potential for those nitrite scavengers. There's also the impact on drug absorption. This is something God reported about at the FDA workshop in June, showing that he was testing 4 drugs, 4 antioxidants, had good results as well, showing that it was didn't seem to impact permeation in his model. And of course, those ingredients have been studied, alpha-tocopherol and ascorbic acid, and have a long history of safe use. Now, with this in mind, we've extended these experimentations internally at dsm-firmenich, and Zretka will share some consideration when working with these molecules. Zretka?
Thank you, Anne-Cécile, and welcome everyone to today's talk. First, I would like to say a few words about the mechanism of action of ascorbic acid and alpha-tocopherol, in specific when it comes to nitrosamine formation. Namely, nitrates, often found as impurities in excipients, will under acidic conditions form nitrous acid or nitronium ion, depending on the pH. Those nitrous acid and nitronium ion will transform further to dinitrogen trioxide and trisonium. Now those two compounds, so they are nitrosating agents, and they will further then react with secondary or tertiary amines in the formulation and form nitrosamines. Both ascorbic acid and alpha-tocopherol will block the formation of nitrosating agents. They will work via redox pathway by reducing the nitrosating agents to non-nitrosating agents, nitric oxide. Now I would like to say a few words about the properties of both ascorbic acid and alpha-tocopherol. Ascorbic acid is actually the best for some moderate to strongly polar environments, while alpha-tocopherol will have more affinity for some less polar and non-polar environment. Ascorbic acid is suitable for various delivery systems, for example, for solutions, syrups, gels, and injections, but as well some solid dosage forms, tablets, and capsules. Alpha-tocopherol is mostly used for creams and ointments, but also capsules and tablets. Here we continue further with the properties of ascorbic acid. And I would like to point out that there are different ascorbic acid powders on the market available. How different? So they differ in their particle size. For example, there are some very ultra-fine powders with a very small, fine particle sizes, and there are powders with a bigger particle size. the particle size distribution is displayed here in the graph. Why is actually the particle size important for the formulation? It will impact the formulation and its stability. For example, you can imagine if for the ultrafine powder, it has a fine, small particles, and it in a way has the big surface area. This surface area is reactive, and since it is reactive, it will have higher antioxidant properties. Not just one property of the powder is important when it comes to formulation. For example, it is also about the morphology. Here we display scanning electron microscopy pictures of the powders I mentioned on the earlier slide. And one can see that the particle shape ranges from the cubical shape to plate shape, but also very irregular crystals. Now, the morphology of the particles, as well as the particle size, is very important for the powder flowability and also powder compressibility. In a way, different physical properties of the powders are important for the tablet application performance. Alpha-tocopherol, in contrast, some physical properties of this compound. It is highly lipophilic compound. It is can be colorless or yellowish brown and viscous oily liquid. It is insoluble in water, but then freely soluble in acetone, ethanol, ether, and vegetable oils. Now, imagine being liquid, one can say it is difficult to include it in oral solid delivery systems, but there are of course ways. So the alpha-tocopherol should be dissolved in a solvent, in this case organic solvent or mixture organic solvent and water, and then it can be, for example, granulated, so added to the solid compounds of the formulation. Alpha-tocopherol is sensitive to air and light. It will fastly oxidize in the presence of iron salts. And then since we know it is very sensitive to air and light, of course, the storage of this compound should be under an inert gas in an airtight container in a cool, dry place. Here further, I would like just to, in a way, mention that there are different manufacturing routes for the oral solid dosage forms. It can be simple direct compression where excipients and API are just blended and then tableted to the tablets. Or maybe some pre-processing step can be included, like wet granulation, where actually you first need to granulate the ingredients, size them, and then maybe lubricants are additionally added, and then tablets are formed. Why we are mentioning this? Because, for example, we have done the study. Here is a preliminary data of showing how ascorbic acid will reduce the nitrite levels. Namely, we have included both manufacturing routes, the ones I mentioned before. So we went for the direct compression and for wet granulation when we produced the placebo tablets, so no API was used. And those placebo tablets contained, so were either controlled just excipients, or they were a set of tablets containing ascorbic acid at different concentration levels, so 0.25%, 0.5%, and 1%. The tablets after production were exposed to harsh storage conditions for 7 days at 40 degrees and 75% relative humidity. And it can be clearly seen two let's say, conclusions. First of all, that despite of which manufacturing route we used, either wet granulation or direct compression, that in both cases, the ascorbic acid did decrease the nitrate content in the tablets. The second conclusion could be drawn is that the higher the ascorbic acid concentration was used, the lower the nitrate content was measured. Of course, this relationship might not be linear, however, the trend is very visible. Another study we performed is actually including an API. It was a model drug formulation that we produced. We have spiked it with nitrate, potassium nitrate, and we have used, in this case, direct compression. There was control tablets, so containing only API and excipients, and there were also tablets containing API and ascorbic acid at 1% concentration. The tablets were then after production, exposed and stored for 2 weeks at, again, harsh conditions. In this case, it was 50 degrees Celsius and 75% relative humidity. And we have tested N-nitroso impurity levels, so API-specific nitrosamines. It can be clearly seen that actually after these 2 weeks of storage, ascorbic acid successfully blocked the formation of this API-specific impurity, while the control tablets showed the increase of these impurities. So here I would like to give maybe a little bit of some formulation guidance when it comes to using the alpha-tocopherol and ascorbic acid, which are antioxidants. It is very important to make use of low nitrate level of APIs, as well as of low nitrate level of excipients, which are now coming to the market more and more, because cumulative effect can lead to higher nitrate levels in the formulation, higher than we expected. But then the use of antioxidants becomes critical as a safety net for this formulation. In case you still have higher nitrate levels in your formulation, adding antioxidants will be very beneficial, because as you have seen on the nitrate level as well as on the API nitrosamine level, it will decrease them, and it will serve as a safety net. We mentioned 2 different possible manufacturing routes for oral solid dosage forms. So bear in mind wet granulation. If you have it in the process, if you want to use it, ascorbic acid can be easily added in your water solution for granulation. And for alpha-tocopherol, a mixture of organic solvent or water, or just organic solvent can be used. When it comes to just direct compression, then one could evaluate ascorbic acid powders with different particle sizes. Wet granulation will pose higher risk of nitrosamine formation compared to direct compression, and this had been already studied and published by Moser in 2022. The instructions are to use low levels of the antioxidants, maximum few percent should be tested. And of course, what is important is to ensure good homogeneity of antioxidants within the tablet. Namely, we want to have a close contact of the antioxidants with nitrate source and API, which will then lead to a higher efficiency. Of course, in case of direct compression, then, for example, ascorbic acid ultrafine powder could be used due to its smallest particle size. Now to concluding remarks, and back to Anne-Cécile.
Thank you, Zretka. In summary, a number of nitrite scavengers are worth investigating, and ascorbic acid and alpha-tocopherol are especially interesting for reformulating strategy and new drug development, as they might have a lower potential regulatory burden due to the safety data around it and well-established use in pharma, nutraceutical, and the food industry. So we keep on investigating these molecules for this particular application at dsm-firmenich, and we are interested to hear about your experience with them and questions you might have. At dsm-firmenich, we have pharma-grade CGMP ICHQ7 quality ascorbic acid and alpha-tocopherol with USDMF and the regulatory support. We always aim to create highly sustainable products with the lowest impact possible on this planet. And we hope you'll visit us at FarmSci in Orlando. We'll be happy to tell you more about this very interesting topic and what we do in this field. But in the meantime, you can connect with us at the contact information right here. And we'd like to thank also René Stemler, our chemist internally, and Marta, who is a formulation expert on our team. I want to thank you for your attention. And with that, I'll hand back to Fernanda for question and answers.
All right. Thank you, Anne-Cécile and Zdravka. Very interesting conversation around this. We have a few questions that popped up here on the chat, and I just want to remind the audience, please put in your questions, and I'll be happy to pass them on to Anne-Cecilia and Zdravka. We have about 10 minutes for that. So, and anything, if you want to follow up afterwards, you will have the contact information from the team here. So feel free to reach out afterwards as well. So I will start with one on the study that we did with the API. And the question is, on that study with the API and ascorbic acid, does the API have secondary or tertiary amines?
So this was a model drug that we used, one anti-inflammatory drug, and it has secondary amines. The structure. Yeah, thank you.
All right. Okay. Another question on when you showed the data on the particle size distribution of the different suppliers and the different grades of ascorbic acid, somebody was asking whether the particle size distribution is a unimodal, is it bimodal, multimodal, if we have any more insights on the actual distribution of the particle size.
So for all samples, we have seen monomodal distribution. In this case, they were quite uniform, but we have seen that actually they quite differ in particle size.
Okay. Keep the questions coming. Let me see another one here. Somebody asked, under what pH range the formation of nitrosamine is possible?
So it should be quite acidic conditions, as we have mentioned. It goes quite too low. I mean, we don't see so much below pH 1 in our formulations, but I guess that would be the strongest. And then it goes up to the 4 or 5. I mean, I don't know, Cecile, whether you want to add more to this, but this is somehow the range where we have seen that it happens.
Yeah, I mean, acidic condition, as you say, is needed there. We've not really studied specifically how much more in what condition or another, but more how, for example, ascorbic acid is active. And there is difference depending on the pH it finds itself. It will be more active at certain pH than others. So not necessarily too acidic, not in the 1 to 2, but maybe more in the 3 to 4.
And what is important maybe to mention, which quite interesting, some questions come up. You are adding ascorbic acid, which is acidic, and you are now saying that acidic conditions should be avoided. It happens to be that actually kinetics, so the rate of blocking nitrosamines is much faster than the formation of the nitrosamine itself. So ascorbic acid is blocking the formation of these nitrosating agents faster. The rate is much faster in orders of magnitude compared to the forming nitrosamines itself, and that's why the whole mechanism works. So don't be afraid of adding ascorbic acid because you think it will decrease your pH.
Thank you.
Of course, unless it's an issue for the API itself and the rest of the formulation.
That is true for pH sensitive, but for pH sensitive APIs, there is sodium ascorbate, which will not decrease, which will be compatible with your API. So sodium ascorbate is for the pH sensitive APIs.
Somebody said that they have used ascorbic acid in the formulation, observed some discoloration of the tablets. And so can you elaborate a little bit more on this phenomenon of discoloration when using ascorbic acid?
Yeah, so it's not uncommon to see some discoloration appear when storing a tablet containing ascorbic acid, especially under high stress, but over time that can happen. So I mean, the browning reaction itself, I mean, you have a carbohydrate, you have an amino acid around potentially, and that's been known for a really long time. And so we know that the browning reaction between the oxidized form of ascorbic acid, because it's a redox couple, it's been observed all the way back in the first observation, I think, dates back from the 20s, but in the 50s they were already looking at this, and in the following decades really looked at those combination. But so ascorbic acid is widely used both as a single component in medicine and its complex pharmaceutical preparation, and no safety issues have been identified. Tablets are also often coated, which can decrease the cosmetic issues. So, I mean, so far we've not seen, at least from the regulators coming in, any issues with the color. But yes, it might happen. And again, the condition it's in, the oxygen, the humidity. There's a number of parameters you can control, and Radka has a lot of information on this, how to make sure you minimize that impact. So limiting exposure to oxygen, controlling temperature, humidity, and the primary packaging, of course, also being important. So it might not necessarily be avoidable completely, but the intensity of the color and the rate of the reaction can be controlled. And if you have an issue with this, I mean, please connect with us. There's a lot of publications and literature that's been published on the reaction itself, on the molecule, which will depend on everything else that's also in your tablet. But to really discuss this topic further, if you want to have more information.
Okay. What were the stability conditions for the nitrosamine assessment? Are they representative of long-term stability at room temperature?
Yes, so you have seen actually for the two studies that we have shown, we had for the nitrate level reduction, accelerated conditions 40 degrees and 75% relative humidity, and this is in a way showing what would happen at room temperature at a longer period of time. 50 degrees, what we have used was more actually to provoke faster. We have seen after 2 weeks, we have seen some results, and they would in a way represent what would happen at long-term stability over a period even of 2 years. So for the longer shelf life.
Okay. Somebody asked about propylgallate. Can propylgallate be used as a stabilizer and as an antioxidant? And then perhaps you can expand to talk not just about propylgallate, but other antioxidants out there, Stravkom.
Yes, so propylgallate is of course also a powerful antioxidant. However, it has been already published in literature, and I think Anne-Cécile listed it at Homschak et al., where they have showed its good efficacy in solutions. However, when it comes to adding it into the solid delivery systems, it actually was not performing the best. So one should always test what is the best for the formulation delivery systems that they have, as well as the API. In specific, there are also publications comparing for other antioxidants. We have learned that it can be, I think as Cecile mentioned about amino acids, for example, when they are compared to ascorbic acid, they also showed in tablets some inferiority. Actually, ascorbic acid was superior in that sense. It is not unusual because ascorbic acid has this high redox potential, which is very important. Besides this, it has a good mobility within the tablet. There are still some air pockets, and also there's some humidity, and ascorbic acid is very mobile and could reach out to the nitrates within the tablet. And then another factor why ascorbic acid is also successful is the solubility. So the solubility of ascorbic acid allows it to faster penetrate through the formulation and act so fast. So that would be the explanations why we see very often in different publications done by different research groups where ascorbic acid shows superiority.
Okay. On the studies that we showed, what were the excipients that were used? What are the other ingredients that were used in the model formulation, and what was the nitrite content approximately of those formulations?
So we have used, for example, for the study with where we tested nitrate levels, we have used some microcrystalline cellulose, also some pre-gelatinized starch, and disintegrant cross-carbamylose sodium. They do have, and we have tested the levels of the nitrate using the latest analytical techniques, UPLC, for example, coupled with MS. The level of the nitrate in all of them is in a range about 1 ppm of the nitrates, and it was already sufficient, as you have seen, to be it was already enough in our study to show that ascorbic acid would lower them to the really low levels and what is relevant.
Okay. We'll do one more question. There is a lot of questions here, so I do encourage everybody to please reach out to us after this. We'll leave the contact information with you on the slides. So let's do one more question, Stacy, if that's okay. Somebody wants to use alpha-tocopherol in a direct compression process. Any thoughts on how to incorporate the liquid into the powder blend process?
So, as mentioned in the description of the alpha-tocopherol being a viscous liquid, it is not so easy to incorporate in a tablet. So, not simple adding to the excipients will not work. It should be first dissolved in a solvent, so it can be used also a mixture for example, ethanol and water. And as such, dissolved alpha-tocopherol can be then granulated with other components. Would be preferred probably to use API in this process also in granulation, so that you ensure the close vicinity of the alpha-tocopherol and the API, and then further after the granulation, tableting can be performed.
And Cecilia,
Thank you
anything else?
all so much. Thank you so
Perfect.
much for a wonderful presentation. Thank you, dsm-firmenich, for partnering with us and creating this opportunity. This webinar has been recorded and will be available on demand if anyone would like to view it again. Thank you to the three of you, and thank you to our participants for being on this webinar. Thank you all very much, and you may now disconnect from the webinar. Have a wonderful day.
Thank you.
Thank you.